Hello, Mr. Allman here, and this is your Allmencast on muscles. Now, you probably think of muscle contraction as being the action of muscles that requires energy, and thus the part that needs ATP. Now, if that's true, why is it that after you die, all of your muscles become taut in a condition called rigor mortis? Well, in this video, I hope to answer that question and explain to you quite a bit more than you ever thought you needed to know about how it is that a muscle contracts. A little basic anatomy first. Muscles are divided into three types. There's skeletal muscles, which are going to be the focus of this video. Those are attached obviously to bones, and that's going to be responsible for all of your voluntary motion. Smooth muscles line many of your organs and are responsible for moving things from one part to another. So you can see that your digestive system works by peristalsis, when smooth muscles are rhythmically contracting to move digested food from one part to another. But also remember that your arteries are lined with smooth muscle, also under the control of your subconscious, and that's responsible for thermoregulation. Finally, you've learned about cardiac muscle, which has a different appearance, a different structure, and it's independent of the brain, contracting based on the action of the pacemaker. Now, the job of the muscles is to contract, but not only that. Muscles also need to relax, and they need to be able to do both of those things at a very specific time. And antagonistic pairs provide a good illustration for why it is that both contracting and relaxing is necessary in order for movement to actually occur. If we look at the antagonistic pair, biceps and triceps, in your upper arm, if the job of those muscles is to move your lower arm, then the biceps, to move the arm up, your biceps need to be able to contract, pulling upwards on the lower arm, but movement will only happen if at the same time the triceps also relax. So the biceps contract while the triceps relax. As a result, work happens and the arm moves up. In the opposite motion, to extend the arm, the biceps need to relax, loosening the grip on the forearm, while the triceps, the muscle on the bottom, is contracting, pulling downward on the forearm. The end result is that the arm is going to move down. So that pairing of muscles, each one doing its job at the right time, is what allows us to move all of these different body parts in two directions. Now, the structure of the muscle has several different layers. The muscle is a bundle of muscle fibers. Those muscle fibers all run parallel to each other. Each muscle fiber, which runs the length of the muscle, is actually a single cell, a single very large cell. The muscle cell itself is divided into different individual fibers called myofibrils. So the myofibril, of which you can see there is a cluster in the muscle cell, is then made up of sarcomeres. Now, the sarcomere is going to be what I will refer to as the functional unit of the muscle cell, because this is where the contraction actually occurs. These sarcomeres are chocked full of protein, two special proteins, actin, which makes up the thin filaments, and myosin, which makes up the thick filaments. So again, you have a muscle fiber, a single cell, which has many nuclei. The muscle cell is made up of a bundle of myofibrils, and the myofibrils can be divided lengthwise into sarcomeres. Each sarcomere is capable of contracting. You can see the striations here, particularly in the image on the right, as you can see those stripes that we see characteristic of voluntary skeletal muscle, and you don't see that same pattern in smooth or cardiac muscle. Now, muscle contraction is known as the sliding filament model. And the filaments are those actin and myosin proteins, and the sliding part refers to the fact that these two proteins are moving past each other with the help of ATP and the nervous system. Now, ATP is required in order to be able to change the shape of myosin. So what ATP is actually doing is binding to this myosin protein and changing its conformation. It's actually changing the angle at which that protein is bent. And that is going to be taken advantage of in muscle fibers to cause movement between the actin and myosin filaments. When ATP is bound, we call that the high energy or active conformation. I will use this image to be able to walk through this process. It starts with a stimulus, which is going to come from the nervous system. When that stimulus comes from the nervous system, calcium ions are going to be released. As they are released, they're going to bind to a gatekeeper protein called troponin. What troponin is going to do when calcium binds to it is slide and move out of the way. As you can see from the second to third picture here, troponin, which was on top, now moves to the bottom of the actin filament. Now, troponin controls the position of a long, thin protein called tropomyosin. Now, when troponin moves down, it's going to pull the tropomyosin with it. Once the tropomyosin is pulled down, that's going to expose the actin filament to myosin. Now, the myosin head here, this is the end of the myosin protein, has an ATP bound to it. And this myosin head has a strong attraction towards the actin protein. So if the tropomyosin is covering it up, it cannot bind. But the second or the nanosecond after that tropomyosin moves out of the way, that chemical attraction is going to cause the myosin to bend down and attach itself to the actin. So as you can see here, the myosin, which is still in its high energy state, is attached to actin. So these two things are chemically bound now. At this point, the ATP is going to be hydrolyzed. ATP is going to be hydrolyzed into ADP and an inorganic phosphate. That inorganic phosphate is going to be released. In the process of doing so, myosin, its conformation is going to change from a high energy to a low energy state. And you can see that it has that additional bend now in it. So the bending of that myosin head is going to move that actin towards the left. It's a very small distance, but this is happening in each sarcomere. Now, if there's no ATP available, then the myosin is going to stay attached to the actin in this conformation. Therefore, no relaxation can occur, because in order for the muscle to relax, the myosin needs to detach and allow these filaments to slide back to their original position. So rigor mortis is going to be a condition that comes about when, after death, because there's no more respiration, no more metabolism, no more ATP available, the muscles are going to remain in a contracted state without the presence of additional ATP to release the myosin from the actin. Now, if ATP is present, then ATP is going to bind again to the myosin. That's going to change the shape of the myosin back to its high energy state, and at the same time release the myosin away from the actin. Now, if calcium is still around, then the myosin is going to bind again. A new cycle will start, troponin will stay moved out of the way, and myosin will bind again, moving the filament even further. However, if the amount of calcium in the system has decreased, then troponin will move back in place, bringing tropomyosin with it and blocking that active site so that myosin cannot bind again. Now, to take a look back at a different image showing the same thing, this is a slightly more realistic picture of what it is that these protein complexes look like. Here is the actin filament, and the actin proteins are represented by the small spheres. Now, winding around that actin and covering up its active site is going to be tropomyosin. And the position of that tropomyosin thread is going to be controlled by troponin. So again, when calcium is released into the sarcomere, the calcium is going to bind to a site on troponin, and the troponin is then going to move the tropomyosin, exposing the active site. So those small pockets on the myosin, that's going to be the site to which actin is able to bind. When no calcium is present, that active site is covered, but when calcium is present, the active site is opened. So what's happening is that the release of calcium is going to be controlled by the nervous system. And there's a spot on these myofibrils called the neuromuscular junction, or NMJ. The neuromuscular junction is going to consist of a synaptic terminal. So we have the axon of a motor neuron. There is a synapse, a very, very small gap between the end of that axon and the myofibril. The axon is going to release acetylcholine into that synapse. The acetylcholine will diffuse across the length of the myofibril and down into small channels known as the T-tubules. The acetylcholine, once it diffuses into those T-tubules, is going to move into a structure called the sarcoplasmic reticulum. And just a quick note on language here, sarco comes from a Greek root meaning flesh. So anything having sarco or sarc in its as a prefix means it has to do with muscle. So the sarcoplasmic reticulum is really just a fancy name for the endoplasmic reticulum of a muscle cell. So the sarcomere is just again, has that muscle root to begin with. Now the sarcoplasmic reticulum is filled with calcium. So as soon as the acetylcholine, the neurotransmitter, is released, travels down the T-tubule and attaches to receptors on the sarcoplasmic reticulum, calcium is going to be released from the SR across the length of the myofibril, where it can attach to that troponin and cause the tropomyosin to move out of the way and allow for the initiation of a contraction. Again, as you can see here, you can see from cross-section the acetylcholine being released. You can see it traveling down the length. You can see that calcium is going to be released by diffusion from the sarcoplasmic reticulum into the sarcomere, where it has that effect. So similarly, there also is going to then be calcium pumps. Those calcium pumps are going to need to use ATP to concentrate calcium back into the SR when the contraction is over. So calcium is allowed to remain as long as the nervous system is continuing to stimulate the muscle to contract. But once relaxation is desired, calcium needs to be pumped out of the way, again showing a use of ATP in the process of relaxation. So the motor unit is going to consist of the neuromuscular junction, which is innervated by these motor nerves, as well as the myofibril itself. Now, that process of muscle contraction is going to be called tetanus, and that contraction is only going to be initiated by the action of many action potentials. So one single action potential generally is not going to be enough to stimulate a muscle to contract. Instead, many action potentials happening in quick succession is going to be needed to cause a contraction of a muscle to occur. Now, muscle contraction is an all or nothing response. So there is no way to be able to have a muscle contract halfway, because either the actin is going to bind to the myosin and cause movement, or it's not going to happen at all. So the way that the nervous system can apply different amounts of force to a muscle is by stimulating only some of the muscle fibers to contract. So if you're trying as hard as you can to pull on a muscle, you're going to stimulate all of the muscle fibers in that muscle to contract. If you only want a small amount of force, then your nervous system will subconsciously stimulate only a few of those muscle fibers to produce an appropriate response. One word on the response of those, so you have muscles that are attached to bones. So you have our antagonistic pair, you can see pictured here on the right. You have the bicep and the tricep. Each of those are going to be attached by tendons to the bone. So tendons provide attachment from a muscle fibers. It's a strong connective tissue that is then attached on the other end to compact bone of your skeleton. Now, you have ligaments as well. Ligaments are going to be another form of strong connective tissue that are going to attach the bones together. Your tendons and ligaments together are going to provide a lot of strength at joints. And a joint is any place where two bones come together. As you can see on the bottom left, there are different types of movement available at different types of joints. As you can compare the kind of movement that you can see, for example, in your shoulder with a ball and socket joint, as compared to what you can see in your elbow. Different amounts and different ranges of motion are available at each of those two joints. Now, in between, your bones are going to have a layer of cartilage, which is a spongier connective tissue, which is going to help to prevent any wear of the bone in the joint. And you're going to have synovial fluid, which is going to provide an important cushioning. That's going to make sure that under vigorous motion, these bones actually don't rub and cause damage against each other. You can stop here at this point if you feel you've had enough. I'm going to go one more time back earlier in the video to review again the process of muscle contraction, particularly in terms of the sliding filament model. If not, thanks for watching. Alright, again, starting up here, the stimulus is going to come as the acetylcholine, which is traveling through the T-tubule, is received by the sarcoplasmic reticulum. The sarcoplasmic reticulum is going to release its calcium into the sarcomere. That calcium is going to bind to troponin. Once it binds to troponin, troponin is going to go through a change in shape that is going to slightly slide tropomyosin out of the way. Doing so is going to expose the active site of the actin protein. Actin is the thin filament, which is parallel to myosin, the thick filament. Myosin has a molecule of ATP bound to it, and it is strongly attracted to actin. So as soon as it has the opportunity, myosin is going to bind there to the actin. At this point, once myosin and actin are bound, we're going to initiate something called the power stroke. The power stroke is going to be when ATP is hydrolyzed, releasing a free phosphate. Doing so is going to change the shape of myosin. Myosin is going to, instead of being bent at a right angle, is going to be at about a 50-degree angle. Doing so is going to exert some force in this image, moving actin slightly to the left. Now, when this happens, the ADP is going to be released. That ADP is going to be released, allowing a new ATP to bind to the myosin. Once ATP is bound to the myosin, it's going to get released from the actin because it's no longer as strongly attracted to it, and myosin is going to return back to its high energy shape. That process is then potentially going to repeat as that tropomyosin will still be out of the way, and that's going to allow for a new binding of myosin to a new active site and a new hydrolyzing of ATP. The power stroke is going to happen. We're going to have myosin moving the actin slightly more to the left in this image, and then the end result is that a contraction is going to occur. Now, this is happening in two directions in every sarcomere, because if we go back to the image of what a sarcomere looks like, back in this image, here again the myosin is the thick filament represented in purple, and the actin is the thin filament represented in yellow. Contraction is happening simultaneously on both sides of the sarcomere. So on the left side, what's happening is that during a contraction, myosin is going to bind to the actin, and the actin here on the left side of the sarcomere is going to move towards the center of the sarcomere, meaning that it's going to move to the right. Meanwhile, simultaneously, myosin is binding to actin on the right side of the sarcomere and pulling that towards the center, so it's going to be pulling left. So you have a simultaneous pulling of the actin towards the center of the sarcomere. And as the end result of that is that you can see those Z-bands on the far left and right of the sarcomere. Now the distance between those has shortened. And so the contraction is going to be the shortening of the muscle as the myosin is pulling simultaneously two actin filaments towards the center of the sarcomere. ATP, again, is used because ATP is going to be bound to the myosin. It's going to be hydrolyzed as the myosin changes shape and pulls the actin. And then new ATP is going to attach to the myosin and release it from the actin. ATP was also used in the process of pumping calcium to concentrate it in the sarcoplasmic reticulum. And again, calcium is going to be used to be able to stimulate the contraction in the first place. If you have any questions, please let me know. Good luck.